This is the third video of the fluorescent and bioluminescent fusion proteins for neuroscience lecture. In this video, we will talk about different applications of the use of fluorescent proteins. One of the most common applications of fluorescence is the use of fluorescently labeled antibodies for protein localization or cell identification using immunofluorescence detection. Immunofluorescence uses secondary antibodies that are labeled with different fluorophores to detect a primary antibody for a specific target protein. In this particular example, the authors want to determine which cell types are responsible for the production of the tumor necrosis factor TNF-alpha. For that, they use three different primary antibodies that recognize three different specific proteins. One is for the protein of interest, TNF-alpha. The other ones are GFAP, which is a protein specifically present in astrocytes. The other protein is NEON, which is specifically present in neurons. What they do is they combine these three primary antibodies and use three different secondary antibodies that are labeled with different fluorophores. When merging the images obtained for every fluorescent channel, we can observe that astrocytes here are colocalizing with the TNF-alpha signal. And therefore, this is the cell type responsible for TNF-alpha production in these experimental conditions, since NEON positive cells do not colocalize with the TNF signal. Other application of fluorescent proteins is the use of chimeras. Chimeras or fusion proteins are proteins created through the joining of two or more genes that are originally coded for separate proteins. Translation of this fusion gene results in a single polypeptide with functional properties derived from each of the original proteins. We can create chimeras with our protein of interest and a fluorescent protein. And chimeras have multiple applications. They are used for studying protein translocation. They're used for studying protein aggregation, protein-protein interaction with the use of FRET, and we will talk about this in a second, and also the use of calcium imaging with calcium indicators. And in the next slides, we will see an example of each of these applications. In this paper, the authors created a chimera between a GFP protein and a SMAD4 protein, or abbreviated as SMAD, which is a transcription factor responsible for the signal transduction of the transforming growth factor beta, or TGF-beta. And by using this chimera, they found that nucleoporin 75 is essential for mediating MAD nuclear translocation, since a NUP75 mutant does not show nuclear fluorescence. As you can see in these images, there is a nuclear localization here and a diffuse signal in the NUP75 mutant. This other example uses GFP fused to the Huntingtin protein HTT. And they show how different polyglutamine repeat lengths stated by Q19 or Q74, which is a longer repeat, affects protein aggregation by a change in the fluorescent pattern in the chimeric protein. As you can see here, there is a diffuse distribution across the cytoplasm, while here the protein is aggregated into small spots. Other application of fluorescent chimeric proteins is the study of protein-protein interactions, and this can be accomplished by the use of FRET. FRET stands for Fluorescence Resonance Energy Transfer, and it consists in the quantum transfer of energy from an excited state of a donor fluorophore to the ground state of a neighboring acceptor fluorophore. what we do to do FRET is we create two chimeric proteins that are fused to an acceptor protein and a donor protein that will only fluorescent when the proteins A and B interact together. These two fluorescent proteins do not fluorescent alone or by themselves, and they will only fluorescence when A and B are close enough within few nanometers of each other. So the donor protein absorbs light and excites the acceptor protein. In this example, the authors developed a new dual color FRET system that enables simultaneous imaging of the activities of two signaling molecules, CMK2 alpha and Rho-A, in single dendritic spines undergoing structural plasticity. Here, you can detect the proteins of interest by individual fluorescence of each other, one in green and the other one in orange. And when the two signaling molecules interact together, there is an increase in the red signal. Another application widely used in neuroscience is the use of chimeras for calcium imaging, or calcium indicators. GFCAMs are calcium indicators created by the protein fusion of an inactive GFP protein, fused to calmodulin and M13, which is a peptide from a myosin light chain kinase. This protein is not active by itself, but when calcium binds to calmodulin, there is a structural conformation in which M13 interacts with calmodulin and causes a structural change in the GFP protein that causes GFP to fluorescence. Here there are two examples in which GCAMs are used to measure calcium. And finally, there is fluorescent chimeras that can be used for cell separation using FACS. FACS is fluorescence activated cell sorting of live cells, which separates a population of cells into subpopulations based on fluorescent labeling. This technique uses a sophisticated piece of equipment that can separate cells from either with labeled fluorescent antibodies, for example, cells that are expressing a specific target, or cells that express GFP chimeras, separating different cells or different types of cells expressing different fluorescent proteins. And within this technique, single cell flow goes through the system and it gets separated depending on their fluorescent properties. Here I put a short quiz to revise some of the concepts that we've seen in class, and then we will talk about more applications of these fluorescent proteins into neuroscience in class.